Hi everyone, thanks for the invitation. I really appreciate it. I'm wearing my Swedish happy socks. And I am very happy to talk about, very briefly, in 20 minutes, gene therapy. When I landed, I was a little bit jet lagged, and so I stayed up watching a Danish show called The Rain, which I don't know if you're familiar with, but it is about the perils of gene therapy, I think. So very topical. So I know that I'm talking to a diverse audience with a variety of interests and expertise, and so I thought I would actually start with a very basic definition of what is gene therapy. And so in essence, gene therapy is the therapeutic delivery of nucleic acids, typically DNA, but not always, into a patient's cells as a drug to treat disease. Kind of obvious. The canonical DNA transcribed to RNA translated to protein means that by introducing nucleic acid, we can ultimately end up with a therapeutic protein which can either replace an absent function of a protein, correct a defective function, or produce a novel function, which is what we call synthetic biology, and therefore confer a function that was never there before. DNA is typically delivered in a vector, which may be viral or non-viral, typically viral. This is one diagram of the possible mechanism of action by which a viral, typically viral vector will confer DNA or convey the DNA into the cell. As you can see, here this is an adenovirus, but it doesn't have to be. It typically isn't these days anyway. So we have the pieces of the viral DNA and in between them the new gene that we want to put into the cell. The vector allows, with hopefully high efficiency, the DNA to enter the cell, is packaged into a vesicle. The vesicle breaks down, releases the virus that will then enter the nucleus and then ultimately lead to the production of a new protein. So I think I took my remit fairly broadly as talking about gene therapy, not just CAR T-cell therapy, although that is what I do. And I'll use two examples, both of which actually from my institution, to talk about both in vivo and ex vivo gene therapy. So in vivo gene therapy is illustrated, for example, by what I will talk about, which is a new drug, if you like, called Luxturna, which is for the treatment of a rare inherited form of blindness. What we do in vivo gene therapy is we actually inject the vector directly into the patient, either an organ or systemically. In this instance, into the eye. So the vector is injected into the subretinal space right here, and then will traffic to the retinal pigment epithelium layer. And then if you enlarge it, that's the same diagram I showed you before. And in that way, the vector and the transgene is conveyed distinctly into the place where it belongs. By contrast, ex vivo gene therapy, which is what part of what I do, is we take cells out of the body, we then genetically modify them, and then we put them back into the body. So in this example of CAR T cells, although you could use this for any other modification of a cell, be it a T cell or a hematopoietic stem cell, a macrophage, whatever, we remove cells from the body. We use a process called leukapheresis, so that allows us to remove billions and billions of white blood cells from the circulation. We then stimulate those T cells in order to enhance two things, really: enhance their ability to take up the viral vector, as well as to give us more T cells that we're able to put in than we took out. In this case, the viral vector is as you can see, obviously put in ex vivo. The cells expand. We remove the beads that were used to stimulate the cells in the first place, since those are paramagnetic beads. And then typically after giving the patient some chemotherapy in order to cause lymphodepletion, which enhances the engraftment of the T cells, we then infuse those T cells back in. So I put together this timeline of gene therapy, focusing on in vivo gene therapy at the top in blue and ex vivo gene therapy at the bottom in red. And in each instance, I tried to, in black, show the year in which it was either first published, or if there is a hyphen there, the year between when it was first published and when it became a reality, by which I mean a high-impact publication, usually followed pretty closely thereafter, actually, by some sort of regulatory approval. So let's talk about the in vivo first. And a particular example that is again relevant to the University of Pennsylvania, where one of the faculty did try to correct a metabolic enzyme deficiency, OTC deficiency. And this is actually quite a notorious case where a patient died because of a very, very strong immune response to a systemic administration of a viral vector. And that kind of, I think, delayed the field for quite a while. There were several issues there, but the field has clearly recovered, as you can see. As far as I could tell, the first approval of a in vivo gene therapy was actually in China in 2003, where pretty simplistically, I would say overly simplistically, a viral vector encoding p53 was delivered systemically. We then have the first published trials followed by regulatory approval, as I'll show you in detail shortly, for Leber's congenital amaurosis, which is what I'll talk about in a little bit of detail. Then we have actually regulatory approval, I think, in Russia for peripheral arterial disease, Glybera for lipoprotein lipase deficiency, actually quite a while ago, and then some really interesting and very, very promising trials for delivery of factor 9 and of factor 8 for for 2 different types of hemophilia. The ex vivo field actually started a little bit earlier with initially a publication and probably not particularly successful, but followed thereafter by very successful trials of ex vivo gene modification of autologous hematopoietic stem cells for an immunodeficiency called adenosine deaminase deficiency at a SCID, so severe combined immunodeficiency, with trials ongoing between 1992 and 2002, and then regulatory approval of Strimvelis fairly recently. I think I will point out at this stage that again the field was set back a little bit by some major issues that occurred from those trials, which were that when gamma retroviral vectors were used to genetically modify those hematopoietic stem cells, there were several cases of leukemia, T-cell leukemia predominantly, that occurred in those children because of integration of the gamma retroviral vector and disruption of a tumor suppressor gene, LMO2. Then there were trials or publications for correction of chronic granulomatous disease, adrenoleukodystrophy, metachromatic leukodystrophy, and the reason I say "or" because there's some children with Wiskott-Aldrich syndrome included as well, where again HSC modification, beta thalassemia with accelerated approval granted, and then leukemia and lymphoma, which is the CAR T-cell world that I will talk briefly about. HIV, I think it is fair to point out, has been attempted to be transmission has been attempted to be corrected by knocking out the CCR5 co-receptor for HIV in T cells, and that's the publication I'm referencing there, and there's work ongoing to try and do that in hematopoietic stem cells, which I think makes more sense. So let's talk a little bit about a sort of vignette, a case of successful gene therapy for congenital blindness. So this is the first in vivo gene therapy that was approved very recently by the Food and Drug Administration in the United States. It was developed just across the road from where I am at the Children's Hospital of Philadelphia by Jean Bennett and Albert McGuire. And what this involves, as I showed you before, is using an AAV, an adeno-associated viral vector, which is fairly easy to work with. It's relatively non-immunogenic. And this encodes the coding DNA for the missing protein RPE65. And so this is delivered, as I showed you, by a subretinal injection. And this is what the trial looked like. This is the latest publication of several. This is a randomized trial of patients who either received or did not receive the bilateral eye injection, and those patients who were randomized not to receive it could then receive it afterwards. And so this is how the trial was done, fully enrolled in 2013, randomization finished in 2014, 2-to-1 randomization. The intervention group first got injected in one eye, then in the second eye, and then the endpoint was at 1 year, and I'll show you some of those clinical output in a second. And the control group, as I mentioned, could cross over to receive this intervention since that would be the ethical thing to do. And the endpoints were essentially related to visual acuity and visual function. And I think this is really important to consider when doing these trials is, do we have a significant endpoint? Is our intervention going to be of use to the patient, not just an immunological response, as perhaps can be seen in some vaccine trials, but is there actually some measurable, clinically measurable and useful output? And there was in this case. So here's a single slide of outcome from this trial. Here on the y-axis were I'm not an ophthalmologist, but this is essentially a way to detect the ability of the patient to detect light. And so the maximum possible improvement is outlined at 6 there with the dashed line. The blue is the control, where you see over time there was no significant change. And then in green is the intervention group. And I have sort of overlaid on this figure from the Lancet paper some possible conclusions that I at least drew from it. And so the first one is there was a significant improvement, so clearly important. The second one is the rapidity with which this occurred. So within 30 days after the injection, essentially the maximal outcome had already occurred. So significant, statistically significant, biologically significant, and quite prompt. And in addition to that, sustained, so for at least 1 year. And this is the reason I'm making this point, hopefully comprehensively, is because this is where the promise of gene therapy lies. So in vivo administration, single administration, persistent, significant, and prompt. So I'm going to switch gears to talk about one of my favorite topics, which is chimeric antigen receptor T cells. And so at many meetings, I no longer give this introduction because I speak at lots of CAR T cell meetings, but I think it is worth to spend just a couple of minutes explaining what this is and why it's so significant. So essentially, CAR T cells, chimeric antigen receptor T cells, are a way of conferring on T cells the ability to recognize something that they normally wouldn't recognize. As you all know, T cells normally recognize antigens presented by the MHC molecules using their T cell receptor. And we have many billions of these cells. But if we want to make the T cells see something that we want them to see, for example, a cell surface antigen such as CD19, a canonical B cell antigen, we have to put that genetic material in there, and that is done by the process that I outlined before. And the canonical design is modular. So it's a single polypeptide encoded by a transgene. And what it encodes for typically is a single chain variable fragment. So this is the business end of an antibody, the recognition domain of an antibody, without the other stuff that is part of an antibody. There is then a spacer which, interestingly, each and every component of this modular design is important. And it has taken 20 or more years to get to where we are today. So the spacer is important. The transmembrane domain is clear. It's important to anchor this in the membrane of the T cell. And then there are this sort of what I think of as a gas pedal, and then a sort of turbocharge. And so the gas pedal is the signaling domain from a regular molecule that belongs in a T cell, which is CD3 zeta. That's normally the molecule by which T cells are stimulated once their T cell receptor binds to the antigen. So for many years, that's all that was put into the CAR construct. But in my view, and that of many people, what really sort of ignited the field is the discovery that the addition of co-stimulatory domains, a sort of turbocharge of the T cells, it leads to superior in vivo results. And this has been very, very comprehensively established. And so there's several ways to do that. T cells have many different co-stimulatory molecules, and one of the questions for the field was which one would work. And so drawing from regular T cell biology, one of the first ones that was used was CD28, and in fact, we still use that one. But at Penn, we chose a different one called 4-1BB or CD137, which is also belongs to a different family of co-stimulatory molecules. Is this important? Well, we know that co-stimulatory molecules are critical for the in vivo function of the T cells. Without those, there were some clinical trials that were very disappointing and had very few people's interest other than the real cognoscenti. So those are critical. Which one to use is actually somewhat of an open question, with the aficionados of the field saying CD28 is, others saying 4-1BB is, and this is an area of active investigation, and it is rather relevant. So once a T cell encounters a tumor cell that expresses that tumor antigen, it lyses the cell, it produces cytokines, it can proliferate, and eradicate leukemia, many pounds of leukemia, and render patients leukemia-free who had refractory disease. So the last year or so has been a very busy time for the field. It's less than a year since the first product was approved by the FDA for treatment of pediatric ALL, and this is what was developed down the hall from my lab. I had nothing to do with its development. So that was for pediatric ALL, and that was licensed to Novartis and has now been approved. And you can see that's exactly what it looks like when it comes to the patient's bedside. Just like that, then is thawed and injected intravenously. Shortly thereafter, a sort of different, maybe even competing product was approved, similar in every way, except it had a different co-stimulatory molecule, a different sort of turbocharge molecule on it, and that was developed initially at the National Cancer Institute by Steve Rosenberg and licensed to Kite, now purchased by Gilead. And very recently, within the last few weeks, exactly the same drug, if you like, the same compound, was then also given approval by the Food and Drug Administration for treatment of non-Hodgkin lymphoma. And so these are really, really exciting developments. And I've had patients treated. I run a clinical trial for a chronic form of leukemia, and I've had patients treated with ALL, with non-Hodgkin lymphoma. In fact, I have one patient who was treated initially with the Novartis product. That didn't work. I just treated him with the Kite product, and well, we have to see what will happen with that. We've also learned some really interesting and novel things from this treatment approach. Cytokine release syndrome. So this is the product label. Whenever a new drug is released, there is a sort of label particularly for important toxicities. And so there's a sort of black label for cytokine release syndrome, which is when T cells are hyperactivated and produce a lot of chemicals, a lot of cytokines that can actually kill patients. And there have been patients who have been killed by this exuberant and hyperactive immune response. We've learned how to deal with that in most cases. There's also another novel toxicity that was never seen before, which is called neurotoxicity, or well, so it is a form of neurotoxicity, which again, we actually don't know what causes it, but is likely related to cytokines produced by the T cells. And that is actually not infrequent, but in the vast majority of cases is temporary. So, and the reason I put that there is it sort of reminds me to talk about the fact that this is actually based on HIV. So we've taken the HIV virus, we've taken all the nasty bits out of it, and we've used it for what it's very good at, which is infecting T cells. And to me, I think when I stop and think about what we've done, this was done initially by Naldini from Milan in the 90s. I think it's remarkable that we've taken something that was and is pathogenic to human beings. We've turned it against its original purpose and used it for our purposes. So I'll try and speed through this because I don't want to take too long, but suffice to say that CAR T-cell for leukemia in particular for childhood ALL, is remarkably effective, with a 90% complete response rate in patients who had failed multiple prior therapies. Really remarkable. The results in lymphoma and the results for adults, particularly with chronic leukemia, are not quite as good. And I don't think we really know the reason for that as yet, but these results are remarkable. And actually, one of the things that I most love about this field is that different institutions doing things slightly differently get exactly the same results, which is very reassuring. So what happens to these patients? Well, if you look at the right side of the slide first, we know that the CAR T cells are actually unable to distinguish between normal and malignant cells that have the same antigen on them. They're very, in fact, I can only think of one cancer-specific cell surface antigen in all of cancer. And so the reason this works so well is because as human beings, we can tolerate our normal B cells being depleted and in fact being gone for many months and years. So this is a Kaplan-Meier curve that shows the probability of having no B cells after infusion of the CAR T cells. So all patients lose their B cells initially, and actually by the time you go out to a year and a half, most patients still don't have recovery of their normal B cells, and we treat these people with injections of intravenous immunoglobulin, so antibodies pooled from normal donors in order to reconstitute their immune system, and most people live very happily with no infectious complications. The overall and event-free survival is a measure that we use in oncology to tell us that the patient has achieved a response and remains in a response without dying from disease or without dying from toxicity, is good, but it's not 90%. So what this shows you is that 90% of children typically enter a complete response. About 50% of them have an ongoing event-free survival, so live on in remission. Very important because these children had no options other than that, but it isn't 100, and so there is certainly more for us to do in this regard, and we can talk maybe later about why it's not 90 or 100%. So just to tell you again, the CAR T cells cannot distinguish between cancer and normal cells that look the same to them. And so while this is tolerable in B-cell malignancies, my own particular area of interest is actually in the myeloid malignancies on that side, acute myeloid leukemia, the myeloproliferative neoplasms. And we have worked out a way of actually getting around that, but I don't have time to talk about that today. So I mentioned the toxicity, which is related to production of these chemicals, these cytokines by the T cells, which has sometimes been fatal, in the vast majority of cases isn't. We like our patients to get a fever. If anyone, for all of you who have a background in oncology, typically a fever in an oncology patient is a bad thing. We expect that there is an infection. That is how people sometimes die from the complications of chemotherapy. But actually, in this setting, our patients and their families are disappointed if they don't get a fever after injection of the T cells, because they know that that is the #1 biomarker for T cell activity. The neurotoxicity is interesting. Sometimes it can be as bad as having seizures. People have died from it, but usually it is sometimes mutism, aphasia, so inability to string words together, as subtle as handwriting abnormalities, and as I mentioned, typically goes away and is an active area of investigation as to why it happens. So I think I have 2 more slides. Future directions of CAR T cells. How do we improve on the basic design? I tried to spend a few seconds actually explaining what the basic design of CARs is. There's still a lot of work to be done in terms of enhancing those and conferring additional functions. And in fact, this is the beauty of gene therapy, be it in vivo or ex vivo, that we can confer and endow our cells with additional functions that may be important. And in fact, one of the things we're doing at Penn right now is, in addition to the CAR, we're also taking out a negative molecule on the T cells called PD-1, which is a checkpoint molecule. So by removing this negative or inhibitory molecule from the T cells, we can make the T cells even stronger. So that's one way we can improve on the basic design. As I mentioned, we're working to extend this beyond CD19 in B-cell malignancies. One thing that we've observed is that, as I think Dr. Hanahan was talking about, adaptive resistance. So one thing that we've observed, which is really interesting, is that children who enter a remission with CAR-19, with this therapy, some of them relapse with leukemia that no longer expresses the CD19 antigen. Remarkable, and was not seen before this potent immunotherapy occurred. So what are we doing now? Of course, we're combining an anti-CD19 and an anti-CD22, which is 2 separate B-cell markers. How do we extend it to other hematologic malignancies? That's something that we're working on right now. And the real gorilla in the room is, how do we move beyond hematologic malignancies to solid tumors? And this is something that we can discuss in the panel session, but actually has proved incredibly problematic for the field. So my second to last slide is, these are the marketed products, the Kymriah from Novartis, the Yescarta from Kite Gilead, and look at everything that's around that. I sort of think of this as, these are 2 pebbles that are thrown into the pond with this enormous waves being created, spreading out from there. And you can see autologous CAR T cells, allogeneic CAR T cells, TCR transgenic cells, non-T cell products such as natural killer cells, macrophages, gamma delta T cells coming out, each one of these being driven by typically a company, sometimes an academic institution. And so, in summary, for gene therapy, there are several products, as I outlined, that have already been approved, and several that are probably close to approval. There are many more coming, as you saw from my last slide. The advantages that I think about are the specificity. I didn't belabor this point, but I think it's quite clear that by modifying or enhancing or removing the function of a single protein this therapy is extremely specific. It also allows us to target previously undruggable or untreatable diseases like this blindness that I was mentioning, the amaurosis, and the really wonderful potential for a single administration with long-lasting effect. I mean, that is as close, if it works well, it is as close to a silver bullet as I think we're going to get. And the disadvantages are the side effect profile. Clearly, nothing is without its cost. I mentioned that people have died from in a variety of different ways, from a variety of different gene therapies. We've learned about how to mitigate these. We've for example, the children who died from leukemia after the SCID therapy, well, that led to re-engineering of the whole process, and then use of different vectors, lentiviral vectors, which appear to be safer than gamma retroviral vectors. There is still, and as a result of those developments, there is still the concern, and I think rightly so, for the development of genotoxicity, for the development of mutagenesis, the causing cancer in the patients. Having said that, through 1,000 patient years of across a variety of different T-cell trials with lentiviral vectors, there's been not one case of genotoxicity. And then I think something that's really important for us to think about is ex vivo manufacturing with its costs and time to manufacturing versus in vivo delivery. And to me, even though that's not what I do, I think the in vivo delivery is much more elegant. Okay, thanks for your attention.
So are there any questions for Saar? I think Aldrin was first, maybe.
Aldrin Utskarpen. Could you say something about the use of CRISPR in gene therapy? How far has it got? What's the potential and pitfalls as you see it?
Yes, there is already a trial in China using CRISPR. We have launched a CRISPR trial as well at the University of Pennsylvania. I don't see CRISPR as different to any other. And one more thing, there will be or currently is a CRISPR-modified hematopoietic stem cell edited for the treatment of hemoglobinopathy, a blood disease. So I don't see CRISPR modification as such as different to any other use of vector, be it vector for introduction of gene therapy. Just because it is a gene editing tool, it has the potential, as do the retro and lentiviral vectors that we put into T cells, still has the potential to cause off-target effects. CRISPR is specific, but it is not 100% specific, and so there is the potential for mutagenesis, essentially. One of my trials that is in development right now is also for CRISPR modification of hematopoietic stem cells, and I think we are all very aware, it's crystal clear that there will be the chance of inducing leukemia in those cells. So to me, the risk-benefit ratio relates to, number one, doing the best you can to have the most specific CRISPR guide, so to analyze exactly where your CRISPR, where the Cas9 is going to bind and cut, what is the potential off-target sites, so not just an in silico screen, but an actual functional screen for where the guide binds that is off-target from where you want it to bind. A very firm and clear discussion with the patient and with clearly the regulatory authorities, and doing this in a setting where the risk-benefit ratio is favorable in patients who have no other therapeutic options and who have a life-threatening malignancy. That's how I see all of gene therapy, actually, not just CRISPR. I really don't see CRISPR as different to any other gene therapy manipulation of the genome in any cell, but we do have to be particularly cautious in hematopoietic stem cells.
Excellent, excellent answer.
Thank you.
Yeah, we'll come back to this in the panel, maybe. There was a question in the back.
Marie Newman, Swedish Gene Technology Advisory Board. There was a paper last year about how the microbiome of the patients could affect the outcome of the immunotherapy, and I wanted to know, what do you think about that? Do you?
Yes, I think that's underexplored and needs to be explored further. Yes, it's an exploding field, actually. Putting hot area of immunotherapy and the hot area of gene therapy together with a hot area of microbiota, that's definitely something that needs to be investigated. There are preclinical and clinical suggestions that is true in a variety of different fields, ranging from allogeneic stem cell transplantation to direct, more sort of targeted immunotherapy, and is something that we're also investigating prospectively, though of course we won't have the results of that for a while.
One more short question. There's so many. Maybe Doug, you can have a word. Can you stand up, please?
Can you elaborate on the gorilla in the room of solid tumors? How do you think that's going to be solved?
You can answer very short, because we can take it also in the discussion if you want to answer very quickly.
Look, I think it's going to be very hard to solve it with the current CAR T-cell approaches, but I'll be happy to elaborate in the panel discussion.
Yes, I think that we will end it here. There's a lot of questions, I know, for Saar. It's a very exciting topic, but we will take questions during the discussion. Of course, you can talk to Saar during lunch. Thank you very much for a fascinating presentation.